Okay, so we just completed our bell ringer. If you haven't completed your bell ringer, go back and do it. So you should have your guided notes pulled up so we can get to it. So these first 2 or 3 slides are just a little bit of review, but it's really important that we've got it down so we can do this next standard or lesson, okay? So I'm going to go through it a little bit more quickly than I normally would, but that's just because we should already know this stuff. So first, blank, the genetic material in cells, or specifically the nucleus of the cell, is contained in a molecule called DNA. And we remember that is that long strand. It looks like a ladder that's twisted. It's got that structure that we call a double helix. It's that spirally shape that looks like a noodle. So your DNA is wound up tightly into that X-shaped structure called a chromosome. So if you see in this picture over here, it looks pretty squiggly. That small line is just a bunch of DNA tightly wound or coiled up, and it forms that big X-shaped structure. Now, on this DNA or on the chromosomes is small sections called your genes. So these genes are, again, just smaller sections. So over here is our long stranded DNA, and here's one section of that DNA is called, we'll just call it gene A, and over here is gene B, and they code for your specific traits. We knew all this. Now, this is the part that we haven't touched on in a little bit, but we just talked about it in our bell ringer. The steps of the ladder of the DNA, that is called a nucleotide. And those are made up of, we call them a base, and it's made up of a sugar and a phosphate group, all kinds of crazy sciencey things that you're going to learn a lot more about in biology next year. But they make up the nucleotide, okay? So moving on to the next slide, looking at those nucleotides when we break them down a little bit farther. And again, I'm going pretty quickly because this is stuff we've already covered and we should already know. There are 4 different nucleotides and they have those long fancy names, but we try to remember them by the letter that they start with, A, T, C, and G, and they are very picky on who they pair up with. So before I even go into that, if we look over here at this picture, this, let me explain it, is if you take that piece of DNA and flatten it out, we are looking at the base pairs here. So we have adenine or A, thymine or T, guanine or G, and cytosine or C. And they are picky about who they pair up with or connect with in the middle to inform those steps of that ladder. Okay, so we always know that adenine or A is besties with thymine. It doesn't want to be connected to anybody else. A and T just want to be together all the time. And same goes for cytosine or C, wanting to pair up with guanine or G. So they're very picky about who they pair up with. A with T, C with G. Okay, so the order that these base pairs line up in your DNA, it's not an accident or a mistake. They are in a certain order because their order makes up that gene. I said that quickly. Let me slow that part down a little bit. My bad. So instead of having your base pairs be at random spots on your DNA, they have a specific order because they are giving instructions for that gene. So it's almost like you're reading a story. Gene A might have a certain story that goes A, C, A, C, A. Gene B might have a different story. It's going to have a different order. So it's going to maybe have an A, then a T, then a G, then a T. It's all in a certain order because it's telling a certain story for that gene because every gene has a different order. Okay, if any of that confused you, please let me know because this was like our very first lesson and I need you guys to know this stuff. I know it's tricky, but again, we keep reviewing it. So if you're a little stuck, please let me know. I can walk you through it, okay? Now, this part is super important for today. Each gene holds information about which proteins that cell should build. We've already touched on that a little bit, but I really want to drive that idea home today, okay? So if you look at this picture right here, let me move myself. Here is our long stranded DNA, and we have 3 different sections of that DNA or 3 different genes. So here we have gene A, gene B, and gene C. Since they are different genes, they're going to have different orders of nucleotides or different orders of those base pairs, right? Now, the order of those base pairs is what is going to tell your genes to make what protein. So gene A has the instructions to make this certain protein. Gene B has totally different instructions, so it's going to make a totally different protein. And same thing for over here at gene C. It has a different order of nucleotides that make up that gene, so the protein that it makes is going to be different as well. Okay, so who cares, right? Why do we care what protein which gene makes? Well, the type of protein that your gene makes for your body is what determines your traits. So gene A might make the protein for, we talked the other day about like facial muscle, so we'll say that. Maybe gene A helps build what your face is actually going to look like. Gene B might help code for the color of your hair. Gene C might make a protein that codes for, I don't know, your eye color. So it's very important that your gene codes for a certain protein and that it gets it right. Okay, again, that should all be, for the most part, review. So let's get into the specifics of what happens if your body messes this process up. Because your body's not perfect, just like how you aren't perfect. So sometimes this whole process of getting a certain order of nucleotides and making a certain protein, a mistake could happen. So what does that look like for us? So anytime you change the number, type, or order of bases on a piece of DNA, that's what we call a mutation. Now, I feel like most of us have probably heard this word before. If you are into Spider-Man, this is a gift from the original Spider-Man. I think it's better than the new ones, but that's just me. We remember Spider-Man, or Peter Parker is his name. He gets bit by a spider, and then he goes through this mutation, and he suddenly has these super spidey senses, and he has all of these superpowers. Same thing with the Teenage Mutant Ninja Turtles. They are mutants, meaning that they have gone through mutations, and now they're massive, super strong, crime-fighting turtles. So mutations aren't this weird, mystical, magical thing. They're real things that happen, but they're not really going to give you superpowers. They're more small changes in your DNA that might change one thing here or there, but not going to let you shoot a web from your hands. Okay, so a mutation happens, like we said up here, when one of your nucleotides or your base pairs gets changed. So if we look right here, this is like half of a DNA strand, right? Because typically it's going to be a ladder, so it looks like we just cut it in half. So we're not looking at the pair of that nucleotide, we're just looking at the one. So here's that strand, and if that strand of DNA goes through a mutation or a change, that means one of the nucleotides is going to be different. So here this T or thymine changed to a C or cytosine. And since your order of nucleotides gives instructions for what proteins to make, this strand of DNA now has different instructions, right? So the protein might be changed as well, which could change your traits. That was confusing, so let me say it one more time. Your DNA strand has a certain order of nucleotides that gives certain instructions for a protein. So if you change those instructions, you're going to make a different protein. And your protein, since it's in charge of determining what your traits look like, if your protein is different, your traits can end up being different as well. So that is what a mutation is. That's what it's all about. Now there are 3 different types of mutations. This looks complicated, I know, but it's really pretty easy when we break it down. They're pretty easy to remember. The first type of mutation is just called a deletion, and that is when a base pair is literally deleted. So here is our normal strand. We had these 4 different base pairs lined up, and these last 3 just got removed. That easy. The next type is the opposite. It is a insertion, when you literally insert or add an extra base pair. So again, here is our original DNA strand, our original gene. And in between, it looks like in between these two nucleotides or these two base pairs, we added in extra nucleotides, added in extra base pairs. So that's going to change the instructions as well. The last one is substitution. I spelled that wrong. Substitution. I can't spell. Okay, so a substitution is exactly what a substitute does. It replaces another base pair. Think of like when your teacher has a substitute, like if I were sick one day and a sub came in, they're not deleting me, or they're just replacing me until I come back. So that is exactly what's happening with substitution. Instead of deleting or inserting, it totally replaces another base pair. So here this C that's paired up with the G, it's replaced with a T that pairs up with an A. So that's also going to change the instructions of a mutation. Okay, so we've been talking about these mutations and how they get changed, deleted, inserted, substituted, but how do they even happen? Like, what's going on? What's causing this to go down? So one thing, and this is usually the most common way that a mutation can happen, is just a random error whenever your DNA is being copied. So we've talked about how your cells are making and replacing old cells by going through mitosis, and that's when your DNA gets copied or replicated and passed down to those new daughter cells. Your body's not perfect, like I said before. Sometimes it's copying and copying and copying that DNA, and it slips up. It makes some mistakes. It says that one nucleotide should be here when in reality it should have been another one. So that is how a mutation can happen, and then that new mutated or new changed strand can be then copied and passed down as well. So all of the new cells are going to have that mutated DNA. So it could be totally at random. The other way mutations can happen is by things called mutagens. So it looks like mutation, but they're gens. So these are factors that cause mutations. Easy. What are these? One would be something called ultraviolet light. Ultraviolet light is just talking about sunlight. So if you spend a lot of time under those UV rays in the sun, you could cause mutations to happen in specifically your skin cells if you're going under the sun. It can cause your DNA to change and then be passed down to those new forming cells when they copy that changed DNA. Another thing that could cause mutations, another type of mutagen, is exposure to different harsh chemicals. So we've seen this sign here that says like caution, do not eat or drink me or be exposed to me. That is because it could cause things to go wrong in your body. It can cause a mutation to happen. Same thing if you smoke. Gosh, I hope none of you are smoking cigarettes. That would be not good. You should stop. But they're bad for you because all of those chemicals you're inhaling are mutagens. They're causing different cells like in your mouth, in your throat, in your lungs especially, to go through those mutations and become less healthy and then be copied down and spread throughout your body. So don't smoke. It's not good for you. Duh. Okay, so last part, and I think this is the easiest part. Your mutations have different effects on your body, right? We just talked about that. It can change your traits. Some of them are good, some of them are bad, and some of them are they're just changes. They don't really change much. Like they're not good, they're not bad, they're just kind of there. So one type of mutation, like I just said, could be beneficial, meaning it's positive. It's a good thing. So an example of that, I am moving myself all over the place today. An example of that is if a bacteria cell has a mutation to resist off antibiotics. So antibiotics are those medicines that you take to get rid of any bacteria in your body if you're sick from a bacteria. that bacterial cell can get a mutation that makes it resistant, meaning that that medicine is not going to be able to take it out. That's not really positive for us, but for that bacterial cell, that's a pretty good thing. It's going to be able to survive and keep living and make new bacterial cells. So that's a positive or beneficial mutation for the bacteria. Another effect of mutations can be harmful. I think this is the one that we know most about when it comes to mutations. So an example of this would be cancer. So if you have a normal body cell and let's say you do smoke these cigarettes, not good, it can cause those, especially in your lungs, those good healthy cells to mutate into a cancerous cell, specifically lung cancer if it's in your lungs. So those new cancerous cells can then replicate and be passed down and spread and just cause you to have some really unhealthy things happening. So that is obviously a negative or a harmful type of mutation. And the last type are neutral. So these mutations are ones that don't really benefit us, like we're not really getting anything great out of it, but they're not really hurting us either. They're just kind of there. An example of that is how we got attached or detached earlobes. You may have heard of this, you may not have, I don't know. But there are different types of earlobes. It's either completely attached to the side of your head, I have attached, see? Or detached, meaning your lobe just kind of hangs off the side. It's not really attached all the way to the side of your head. So this happened from a mutation a long time ago, and that person was born with detached earlobes, and then it just got passed down and passed down and passed down. So we don't really get anything good from having attached or detached earlobes, but it doesn't really hurt us either way, depending on what we have. It's just kind of there, right? So it's neutral. Okay, so that was a lot today, so let me go over it real quickly again, and then I'll let you go. Remember, we talked about how our DNA tightly coils up to form that chromosome, and on our DNA, we have those small sections called genes, and the order of the nucleotides or the steps on the ladder of DNA is what determines that gene. And your gene codes for a specific protein, and your protein determines your traits, such as eye color, hair color, whatever. A change in the order of those nucleotides is called a mutation. So if you have a mutation, those nucleotides are changed, and that can change the protein that's being made, and it can change the trait. There are 3 different types of mutations. Deletion, when those base pairs are literally deleted or erased. Insertion is when you're shoving in a new base pair, or substitution is when you are replacing a base pair. Different things can cause those mutations. It can either just be your body screwed up when it was copying its DNA and it made a mistake, or you are exposing yourself to different mutagens. Maybe you're in the sun for too long, or you're being exposed to different harsh chemicals. Once you get a mutation, they can be either beneficial, meaning positive, meaning it's a good change, it's going to help you in the long run. It can be harmful or negative, meaning it's hurting you, or it can be neutral. So it's not really hurting you, it's not really helping you, it's just kind of there. Okay, if you have any questions about anything we talked about today, hit me up. If not, I will see y'all tomorrow.